We see one pattern that is common, and I must mention also that here that we took just the domain that harbored the beta-lactamase to further go down. So to help us understand it better, there is a high conservation at this very domain with humans and mouse and also across the different plasmodium. But then one thing that is very common and has been shown is conservation of some residues that have been shown to enable this domain to bind to divalent ions. So we mapped it, and clearly those residues were conserved. So for us to understand it, we have to go into the parasite. So we made use of this plasmid which is available. It allows us to target with a triple HA and then follow. We chose HA because HA is small and it wouldn't have any interference with it, whether it is exported or not exported. So it allowed us, we got the gene tagged, and we're able to show that the gene, as we have really thought it, got integrated into the genome and is at the right size. The next thing, once you have it, is to go and see where is it expressed. So we took the different stages in the asexual life cycle. From ring stage to schizoid, we see it well expressed. So we went to also confirm that with Western blots. Then we went to the sexual stage in the gametocyte, and it is very nice expressed through the different stages of gametocyte development. So we sought to now look further, but on this note, we have come to understand that, yes, this gene is expressed in all the stages as pertaining to sexual and asexual life cycle is concerned. So we wanted to look at where is it localized. So this is a 3D sim, but this is just a slide of the 3D model we tried to do. And much as this tells us the resolution difference between these two, this is a gametocyte-specific gene, protein. We don't see any colocalization, but if you use the normal wide field, you will say that they are colocalizing. But if you go down at a higher resolution, that isn't what is happening. So the next thing, having known that it harbored the beta-lactamase domain, we wanted to know, what does this do and what is so unique about it? These are enzymes that catalyze or they help us synthesize NAEs and also phosphatidic acid. So just last week, somebody presented in the conference that phosphatidic acid is necessary for invasion. So I've not drifted so much from invasion. So looking at this, it gave us the lead. And one of the enzymes that harbors the domain is the NAP, that is N-acyl phosphoethylamine phospholipase D. So we went back into blasting again to see whether our gene really goes with any of these enzymes that are up here. And clearly, we find it to have higher similarity to phospholipase D in different organisms. Because of time, I'm not showing that much. And we looked at what are the functions of these in different organisms because it's wide conserved. So in animals, it has been shown to have a wide range of functions. And one thing that is very common to whether animal or plant is the ability to stabilize membrane. So indirectly, it could have something to do with lipids on the membrane or modulate those kind of mechanisms. So it gave us another platform. If you could see this domain, then another tool that is available in the scientific domain is well characterized and also well genetically. With yeast, there are a lot of genetic tools to really go down and find the functions of the gene. So we sought to see if this gene is really the enzyme, then if you go to the yeast, we should see activity. Because yeast, homologs of this gene has been shown to be not essential. So we went into the yeast, we designed these two constructs, and we designed these two constructs because earlier in yeast assays, they showed that you will only see a phenotype if you knock out the homolog and also an upstream enzyme that acts on the same pathway. So we knocked out the 2. So we had a total of 10 constructs, and in all we had an empty plasmid as a control. So one thing we show is that if you just knock out, this is the wild type, you knock out just the gene we are looking for, you don't see any strong change in phenotype. But if you do a double knockout, you see a strong effect. And if you double complement using Plasmodium, you see a rescue of the effect to some degree. So we further went on to do a growth assay for these isolates to see if really the function is that conserved. So with all the 10 constructs in an assay where we measure the OD of the growth of the yeast, we could see as you knock out the 2, you see a strong decrease in the fitness of the yeast and its growth. So as you complement, you slowly restore the function. And this is being shown here clearly. So what this tells us is that now we are likely characterizing Plasmodium falciparum and its alpha-phospholipase D enzyme, which helps the parasite in one way or the other in carrying out some function. So the other thing is to see if it's functional, then can we purify it and see also in the test tube, will it really give us an idea? So we clone this domain of the protein, which is 57 kilodalton, and fortunately for us, it had formed a complex. And why this complex? Because it had been proposed in other studies that beta-lactamases have the ability to oligomerize up to 20. So we went to see, and this was heptamer, and clearly we could do a lot, but we did a knockout in the parasite, and we confirmed that it is really knocked out, and we showed that in growth in the parasite, the growth rate decreases as you grow, you increase the number of cycles of the parasite development. Then also to look at the phenotype, you see that there is a kind of enlargement in the parasite as they progress. At the ring stage, you don't see any strong phenotype, but then as they progress, you see an enlargement, which we have not yet characterized, but we think it is the food vacuole that grew out of proportion. So with that, we just